So I'm going to talk about the topic. So this year, the talk is not really fully theoretical, or it's not really a pure theory talk, and it's going to be, but it's going to be really informed by modeling. All the analysis that I'm going to show, it was really done by Jose Valdez, that is a postdoc here in my group here at iDiv. So this is work that he really led over the last year, and that we published in Ecography earlier this year, that I'm going to present. But this addresses a very, very important topic, that is, can we measure what is happening to biodiversity? What do you know about what's happening to biodiversity? I've mentioned this in the previous talk I gave here at CBU a few years ago. And so the basic context is similar, but today I'm going to go in a new direction based on this new work that we have developed over the last couple of years, and that we are still developing in reality. I'll talk a bit more about that in the end. So I want to start by what we think we know. Maybe there's 6 mass extinction, although many people have questioned if this is really the 6 mass extinction. Certainly, there are no shortage of papers, and even the consensus from the IPBES report is that we have something between a crisis, a huge biodiversity crisis, and a mass extinction with 1 million species threatened. Between 1 and 2% of the species already lost in the last few hundred years, and more recently, all this work on population trends of insects have been also catching attention. In reality, the first study that caught global attention was done here in Germany by volunteers, and it was a complete surprise when they found major biomass declines in these traps, in these insect traps, in protected areas. So it was not even agricultural landscapes. So the effects were really surprising. So it left to the idea of an insect apocalypse, an insect Armageddon. I love this cover from Design. At the time, Angela Merkel was still the chancellor, and it says the silence of the politicians. This had a huge impact, for instance, in the politics, both in Germany, but also at the European level, with a lot of measures being taken to study more these phenomena, to study how it can be addressed. And in reality, at the European level, I would say that the European biodiversity strategy, the part of the new Green Deal, and even the ecological restoration law that was just passed in the parliament this week, in a very funny or a very interesting process that is not over yet, is also a bit an offspring of what people came to realize, that there was also a problem with the insects. And the other, let's say, big trend that people have looked at is the trend in vertebrate populations. Particularly, there are 2 major indicators people have looked at. One is the so-called Living Planet Index, that is the average population growth rates across population around, over 10,000 populations of vertebrates around the world. And there is also indicators based on population trends of birds, both for North America and in Europe. We have the common farmland bird indicator and the common forest bird indicator. And also, we also have for butterflies. But for the birds, the thing that happened that caught really attention of the public and the media was, we have known that they have been declining. The number, like 30% declines in Europe, has been around for many years. But a couple of years ago, a paper in Science translated this into number of individuals. And they said it's 3 billion individual birds that disappeared. And this really took the media by the storm. And And so this is, let's say, what many of us know about biodiversity loss. But on the other hand, several people have questioned this paradox. People like Chris Thomas that has argued that like invasives are increasing reality species richness in many regions of the world. And this idea of this biodiversity conservation paradox, that despite all these extinctions, in reality, things in many places are going better. And mainly these two papers really, let's say, are what start this paradox. Is this paper published by Miguel in Science in 2014 and by Mark Velland and colleagues in PNAS in 2013. And what these papers looked at was trends in local species regions. So the first paper from Mark Velland does that for land plots. So you have a plot, let's say 1 square meter, you go there and you measure species richness. And then you go there after 30, 40 years, 10 years, and you see, do you think species richness in the plot increase or decrease? And they found, to the great surprise of the scientific community, when you do a meta-analysis of this kind of plots that have been resurveyed around the world, there's no trend. You cannot find any trend. Some have increased in number of species, some have decreased, but the mean is 0, or not detectable, not significantly different from 0. And the very similar result was found by Marie Dornelis when they built this database called now BioTime. And what they did was they tried to get all kind of community assemblage data sets that they could that have been resurveyed at least 2 moments in time. It can be more, but at least 2 moments in time. And they tried to get all kind of transects from plankton surveys in the ocean to mammal surveys let's say in the UK, all over the world, as many assemblages as they could, and look at the trends in species richness over time. And they found that there was no trend in species richness. And so there's been a big discussion on what this means. In reality, there's, let's say, almost 2 fields in biodiversity science right now, 2 fields of people, or 2 camps. And there is the people that say all these studies are bullshit and there's so many problems and you cannot trust them. And the other camp, they say, no, we stand by our science is rigorous, this is what's going on. The narrative of biodiversity status in the planet needs to be rewritten, and we just haven't gotten it right. And these 2 camps of people have been really in a heated discussion. We brought the 2 camps to a workshop in iDiv a few years ago. I was unfortunately away, I was not involved in that workshop, but there was a workshop here with people from both camps. And it was called the exchange workshop. And in the end, some people ended screaming at each other, and there was it was kind of not possible to have a consensus dialogue with everybody in the room. So that's how heated the discussion is. And today I'm going to tell you what I think is the problem with this data and why both camps, let's say, have some reason. And what you need to understand is why both perspectives, let's say, have some scientific merit, but there is really one answer about what's going on. And the first thing I wanted to highlight is that these results were very surprising because people thought that if you look at the first numbers, I'm going to look at the slides, again, the first slides here. If you look at these slides and you see, okay, 6 mass extinction, 1 million species threatened. You look at this slide, 75% decline. You look at this slide, global wildlife populations plummet by 2/3. This is the Health Living Planet Index. And you look at this decline of North American fauna, 3 billion birds lost. You immediately got into the perception in the public or as a scientist that there's a massive decline in biodiversity that must be everywhere. Is it, if the first impression that you'll have is that. In reality, this perception was so strong that an entire big research field has developed over the last 30 years called the biodiversity and ecosystem function research. It's something that I guess there's not a lot of people in CBio working in biodiversity and ecosystem function research, but it's, for instance, very big here in iDiv and very big in many, many other research institutions. In Montpellier, for instance, and our partners in BIOPOLIS, BEF is pretty big. I don't know if there is anyone here in the room that has interacted with colleagues in Montpellier working on BEF, but they have a mesocosmos experiment also there to look at the relationship between biodiversity and ecosystem function. And all the studies they have done, typically you manipulate species richness and you try to see how ecosystem function is changing. And all the studies that they have done, they have assumed that there's major declines in species richness in ecosystems, and that is affecting ecosystem function. Primary productivity is reduced, or stability of primary productivity is reduced. And so there is a lot of 30 years of research, an entire field that is based on the assumption that species richness is being reduced everywhere in significant numbers, because it's not enough to go and remove one species from an assemblage of 40 or 50. They knock out serious number of species, like knock out half or 2/3 of the species to see effects. So it's not like enough just to go there and change one species in the plot. To see experimental effects in controlled settings, you really knock out a lot of the species. So there was this belief that, okay, this is what is happening in the ecosystems. You are losing the species, what's happening in the ecosystem function. And then when you start finding out that you're not seeing these massive declines in species richness in local ecosystems. Yes, maybe there is new species coming in, maybe generalist, maybe there's homogenization, maybe you need to look at the identity and the traits. But you even have to question, was this BEF research where thousands of scientists have worked on over the world and I don't know, hundreds of millions, billions of euros were spent in this research? Was it, did it make sense as a management of biodiversity research, given that the assumption that big declines are happening may not be true? But the issue is with I'm digressing here just to say there was this perception that was even based in informing some of the research. There was massive declines in species richness, and we need to look at ecosystem function. And this perception is very likely wrong. And the reason for that has been in some of my work before, but in the most recent, the most recent work that we did here, one of the most recent work was with Ingmar Stod in global ecology and biogeography. And we looked at this. This is a difficult thing to do, but because there's very little large-scale data, let's say landscape and larger data on extinctions, spatially explicit. But what we tried to do was we tried to combine information from land use change from the 20th century, from the 1900s until present, and we looked at large 1-degree cells around the world so around 100 by 100 kilometers. And look where you had lost habitat in this period. And we looked at how many species you lost. Now, we don't know that answer, that scale exactly. There's no surveys that have been carried out over the world at this scale 100 years ago and now. So what we did was kind of constructed surveys based on GBIF data. And it's a very iffy method. Yeah, you need to believe that GBIF data is sufficiently abundant to do this. There's the problem of, you may have observed something in 1900 that you don't observe now because nobody went there and checked if the species was there. So statistically, this is a bit iffy, and we have to try to come up with a couple of ways of addressing, let's say, uncertainties in the data. But when we did our best shot in doing this analysis, what we realized is the following. Even when you lose all the native habitat at large landscape scales, you lose very few species. You lose between 10% and 20% of the species. So it means that the majority of the species are able to persist at least for some time in landscapes after all the landscapes have been converted to human-dominated uses. And so this poses immediate problem. If this happens in the worst places where you still have 90% of biodiversity, this is the probability of persistence of the species in the y-axis and the amount of habitat loss in the x-axis. If you have up to 90% species persisting, and it's interesting that not all species are equal. Species with larger ranges, the ones that are kind of invading around the world, persist more than species with narrow ranges. This is very interesting.
Não posso fazer nada nesta casa.
And so the narrow range species, 20% of them go extinct. In contrast with the large range species, only 10% go extinct. So there's different vulnerability of large range and small range species. And this is kind of a double vulnerability because the small range is already more vulnerable because they are to habitat loss because they are small range. But not only that, in the same unit of size of landscape transformed, they are also more vulnerable. They have some kind of trait that makes them more vulnerable to habitat change. But a lot of them persist. So maybe part of the problem is that the size of the expected size of the biodiversity crisis has been kind of I dare to say exaggerated. Yeah, there is loss of species, but it's not as strong, even in the most degraded landscapes, as one might expect from reading some of the literature or from reading some of the media. Yes, there is biodiversity loss, but it's not as intense. And that brings the problem of, if this biodiversity loss is smaller, could you still then detect a trend? And that really poses an issue. And so we went to do this thought experiment on this. And the thought experiment was based to address this question: can current biodiversity monitoring efforts accurately detect global biodiversity change? How this can be affected by sampling biases and measurement errors? And the idea here is that you start with some representation of how biodiversity is changing. And again, this is part of the problem, because if you are assuming that biodiversity is declining 90% in the last 100 years, it's very different if you assume that biodiversity is declining 1% at the local level in the last 100 years. So you have to start, in our thought experiment is, can we start with some realistic values for biodiversity loss? And can we have some spatial, because we want to do this as a spatial analysis to look at spatial effects, autocorrelation, spatial effects, and so on, can we have some spatial estimate of this biodiversity loss over time? And so to do that, we don't have the data, we don't have biodiversity trends measured globally, spatially explicit in the world directly, but we do have data sets that can allow us to do that. And one is the predicts data set and the predicts model. And we use the predicts model as the real world. So we assume that the predicts outputs of how species richness is changing over time in each degree cell around the world, in each 1 by 1 degrees cells around the world. In reality, we go a bit higher resolution, but we come back to that. In each cell, grid cell around the world, we know what has been the species richness trend from predicts. We assume we know. And then the question is, we know it in, let's say, in the baseline year of 1980, in 2000, in 2007, 2009, and 2010. So we did this in different years. And then we say, well, let's say we know that we know hypothetically how biodiversity is changing. It's not this 90% loss. I'll show you in a moment how much it is. And now let's say that we put a monitoring network of a few sites randomly placed around the world, and you count the number of species in those sites in the base year and in the resurvey done in 2010 in this model. If you do that in 100 sites or 500 sites or 10,000 sites, can you find a signal in the species richness? And we do a very simple one sample t-test. I'll show you in a moment. And when we ask, is mean species richness change of such a monitoring network of sites significantly different from zero? So you get 1,000 sites or 5,000 sites. What is the mean change in species richness in each site as detected based on the predicts projections of how biodiversity is changing, how is the real world changing. And we do many random monitoring networks. We do 1,000 random monitoring networks, and we see in how many cases we find that there is a negative trend in species richness. We did different scenarios, as I said, varying the sampling interval, the spatial grain. If you have very high resolution samples, or where you measure in a small plot, or you measure in a very large plot, in a landscape plot. We'll look at the effect of sampling bias, for instance, biasing towards most studied biomes, or excluding areas with a lot of loss. And we look at the effects of measurement error. So let me tell you a bit more about predicts. You may have come across this data set. Originally, it was compiled by Tim Newbold and Andy Purvis and Georgina Mays and others. The project is called Projecting Responses of Ecological Diversity in Changing Terrestrial Systems. And what they did is they compiled a huge database of studies where people have measured species richness in native habitat and in human-modified habitat. Let's say agricultural, forest management, grassland, urban ecosystems. And they compiled a huge database, and they calculated the average. They kind of did a GLM, a simple model that tells you what is the average species richness loss when or gain. Sometimes there are gains in average for the species richness when you do a conversion from a native habitat to some other human-dominated use. And then they use that to combine with the land use map to project how biodiversity is changing over the world. And doing that also over time, you can have also the species richness change over time. And the result is that in the last 30 years, and this is an important number to have in mind, in average, across all the grid cells of the planet, you have minus 0.5% reduction in species richness. That's really small. Okay, it's half a percent point of change in species richness. So the challenge out there is, if you want to detect global trends in local species richness, if that's even a meaningful thing, because in some areas of the world, species richness is increasing, other areas may be decreasing. But let's say this is a meaningful thing that you want to detect. You have to be able to detect a 0.5% change over a period of 30 years. And if you go for a period of 1 year, of course, it is a much smaller change, or a few years. So can you do that? In what conditions can you do that? And that starts to, this starts to kind of sink in this challenge of detecting trends in global trends in biodiversity from local species richness, because it's such a small trend that you are about to detect, and not the big trend that was in many people's minds. So we did this analysis, and let's start with the first question. How many of these networks, or what in average, can you detect a significant t-value that tells biodiversity is declining? So in the x-axis in this figure, you have the number of monitoring sites. So networks of monitoring goes from 100 sites to 10,000 sites. And you looked at these 4 time intervals. 1 year, just looking at 2009 to 2010. That's a very small biodiversity trend to detect. 3 years, 10 years, and 30 years, it's about the 0.5% I was telling you. And this box plots tells you the distribution of the t-values, and the significant t-value for difference between 0 are ones that with gray bars, so around t-value of -3 is significant in this test. And we see what is, when can we detect these global trends that we know exist because we have modeled them. We are using the predicts. We know that they are negative. We know what's the value. We're trying to say, can we detect a negative trend or not? In reality, even a positive trend, some trend different from 0. And what we find here is that, for instance, if you just use very short time series, 1 year, it's impossible to detect with even with 10,000 monitoring sites. You could extrapolate. It's very, very hard to, with any number of monitoring sites, to detect in such a short time span. The situation gets better for longer time series. And in theory, if you have 30 years and you have 500 sites, you could detect, in most cases, a significant trend of local species richness. And with 10,000 sites, you are likely to detect even with short time series of 3 years. So these are the good news, isn't it? So if you have 10,000 sites perfectly monitored over 3 years, you could detect trends in local species richness. The problem is that these 10,000 sites don't exist. We don't have data from 10,000 sites being monitored in a consistent way every 3 years. So it's an hypothetical question. Not only that, this is assuming some perfect conditions. And so what we did next was we added a bit of error, measurement error. You know, let's say it's not the same observer that went there in the first survey, in the resurvey. But there is some error. You know, if you go and sample a plant plot, for instance, you may not count the same number of species twice because you missed some in one of the counts. Or if you go to do a bird survey, there is variability. day-to-day variability, and you may not count the same number of birds and the same number of species. So if there was no error, and if you have a few years of time series, you could detect the statistical power to detect a negative trend is very, very high with a few number of monitoring sites. So that's the red line here. If you add 1% error, you're still doing relatively well. But as soon as you have a 2% error, and it means 2% error is that if you have 100 species in the community, maybe you just counted 98 in the first round, and in the second round you go and count 99. Well, 2% error there. It starts to be difficult to detect any trend. And if you have a 5% error, and there is good, we looked at this empirically, and there is good evidence that most estimates of species richness from community time series may have errors greater than 5%. If you have 5% error, you just don't detect any trend for any number of monitoring sites. So a very small error of 5% will not allow you to detect any statistical trends on local species richness. That will explain, of course, Meridionell's results and Valens, marked Valens results on their teams, if there was some measurement errors in the community assemblages over time. We also looked at the effect of spatial grain. This is a really interesting one because, in the top figure, we have what the predicts model is telling us. And in the left, we have very fine spatial grain. So we have very high resolution cells. And in the right, we have coarse spatial grain, where we have done the average value of the neighbors, of 9x9 neighbors. Yeah, so that it kind of smoothes the signal of biodiversity change. And the colors is how much species have changed. So in some cases, you have species richness increases. This happens, for instance, when you have abundant farmland or abundant pastures, for instance, in the tropics that have been renaturalized, and you see the species recovering. So that's the blue areas. No change, the yellowish, and declines are reddish. Okay, and then in the bottom you have the histogram of the cell distributions of values, and that's very similar to the Valent histogram that I showed you before, where in most cases there is it seems that you have some increases, some decreases, but it's very symmetric around 0. It's not fully symmetric, but it's very symmetric. Now, what is interesting thing here is that if you smooth it, if you have a coarse spatial grain, it's easier to detect a negative trend in species richness or any trend in species richness. And the reason for this is because you kind of average out the noise in the signal. Yeah. So if you have very fine spatial grain, you really need to hit the places where there is losses. Because if you sample, this is kind of what we call the parking lot problem. A lot of the time series we have are from places that have not been heavily degraded over time. If a place, for instance, is converted to a parking lot, nobody goes there and goes and counts plants again. Who's going to count plants in a parking lot? It's not going to happen. So if you have these kind of situations, you really need to be ensuring that the parking lots are still being sampled. And if you have a very fine spatial grain, if I sampled 50 meters away from the parking lot, maybe there's no signal. Maybe the community of plants is still doing very well. So it needs to be you need to be really sampling in the parking lot. And this is why if you have kind of a smoothing of this signal, you can more easily detect a negative trend in biodiversity. Now, this has implications for the type of monitoring that you do and for the taxa that you monitor. So let's see how this has implications for the taxa. If you do plants, they don't move around, so they move very little through dispersal. So the parking lot problem is the biggest problem, because if I don't sample the exact plot that has been degraded by habitat loss, I may not sample any change in biodiversity. In contrast, if you have a mobile group like birds, mammals that move around in the landscape, when you sample anywhere in the landscape, you're not sampling only that point in the landscape, you are sampling that point and the areas around that the birds or the mammals are using. So this is an argument. I'm sorry for my plant colleagues, my plant ecologist colleagues, but this is an argument why monitoring taxa that are highly mobile, like birds or mammals, may be more providing better information about biodiversity loss, because this taxa is doing the averaging of the landscape for you. It's doing this coarse spatial grain for you. Another alternative is to do it by the monitoring design, where you ensure that you monitor larger areas, larger sampling plots, not the 1 square meter plots, but you monitor square kilometers, and large areas need to be monitored then. That will also solve the problem. So you can solve this problem by either choosing a taxa that does the spatial averaging for you, or yourself doing spatial averaging. And I wanted to show you, maybe, but in this view is not so clear, that in the distribution of the histogram, what is going on the values. So in the bottom left, you have the most, let's say, the values are the biggest range of values is there, but it's not very very clear. But you'll have to believe me that you have the standard deviation of the values is the biggest one in the fine spatial grain, and the standard deviation is the smallest. I think you kind of see it slightly that it's hard to interpret this figure in the way that I wanted to show you. The standard deviation is the largest in the left figure, and the standard deviation is the smallest in the coarse spatial grain. And because of that, your estimate of the mean is more precise, even if you don't have the extreme losses. Like you have here, the red areas, you have extreme losses. These are smoothed out when you smooth the landscape scale. You don't have, if you do the smoothing, you don't have any places where you have extreme losses, but you have everywhere a bit of loss. And that everywhere a bit of loss makes it you can detect a stronger negative signal than if you have the high resolution data. Okay, the other thing you want to look at was a kind of spatial bias that you can have in the data. And it is well known that a lot of the sites that we have data from come from EU and US. And that sometimes these places that have been really degraded are not resampled. What is this effect of spatial bias? Well, if some spatial biases may be as big that we would even detect a positive trend in local species richness, although the global trend is negative. So for instance, if we just look at the United States and the EU, you may find a positive trend in local species richness due to farmland abandonment, and increasing species richness around the world. In the abundant habitats. And so this will not provide really a global picture of what's going on. If we exclude the biggest losses, the 1% biggest losses, that's kind of the parking lots of the world, you may also have very difficulty detecting the trends. And if you exclude the top 5% losses, then the trends become potentially positive. So any kind of spatial biases would really give you the wrong answer. Okay, so what are the main results? The local biodiversity for detecting global trends is highly sensitive to time series lengths, sampling or observation errors, let's say observation errors, the resolution of the monitoring or the mobility of the taxa, and spatial biases. And so we may fail not only to detect the right trends, the correct trends, we may even find the wrong trends due to some of these biases. So how can we fix this? How can we improve our analysis of biodiversity trends? How can we design better monitoring? Well, first we need to be very careful in designing monitoring sites that are representative spatially across ecosystems, across regions, across levels of drivers you want to examine. And you need to emphasize the repeatability and the high quality of the monitoring on those sites. We don't have such a network. We have now a project, EuropaBond. You can go europabond.org and check the ongoing project. We are doing this, designing such a network for Europe. But we have to go beyond just looking at meta-analysis or synthesis of data of time series. This is never going to get you the right answer. You need to combine that with model data, like the predicts have done. And so we need to increase the use of models in the analysis of biodiversity trends. Nobody just goes and measures climate change just with temperature stations. That's not, the way that you do that. You use a temperature station to calibrate biodiversity climate models and to then project global changes in land surface temperatures or sea surface temperatures with models. It's not only the time series. It's the same thing for biodiversity. We need to use models to correct for the bias that exists in the data and even bias that may exist in well-designed monitoring networks. So this is what I had to share with you today, and I'll be glad to answer some questions. Thank you.